Okay, hello, class. Um, Something has come up. Long story short, I was given not one but two different schedules that were both incorrect, and I will, in fact, be at the hospital this Sunday. And I found this out a few days ago. I've reached out extensively to Varsity Tutors and have not been able to find a solution with them. So I'm taking things into my own hands. There's a protocol to get a substitute, and I was willing to meet with a substitute and make a lesson plan for them, or was let them wing it, do whatever. Y'all might have liked that person more than me, and y'all could have fired me or nothing. I don't know. But the link that normally appears on the website to make the substitute request was not there. Did not see it at all. I sent an email, and they told me to click that link, which wasn't there. So I've told them that. They told me to follow up with this other email. I sent them an email and have gotten no response as of yet. And being that it's where I live, it's 10:30 on a Friday night. I don't think I will get a response before Sunday. And even if I do, I'm just going to rock with this option. If for some reason they come through with a substitute on Sunday, feel free to go to that lesson, and this will just be bonus. Okay. However, if they don't, this is what you've got. I doubt we go an entire hour and a half on this lecture, because built into my time when I decide these things, there are time for questions, time for y'all to respond, and I try to spend at least 15, if not longer, more like 20 or 30 minutes answering y'all's questions and discussing with y'all. And I want y'all to give answer things that I say. And I actually would like to be a little bit more active now, maybe use our speaking voices a little bit more in the chat or in the lessons, but that's okay. But I know last time we had a couple out loud questions that I took a long time to explain, but there is more merit in me directly addressing your concerns and your needs than me planning a lesson and just sticking to it to a T. So for that reason, I want all of you to email me as many times as possible, as many times as is needed, to get through your questions for this week. Okay? I need y'all to, you can do the group me and ask me questions. You can do the email and ask me questions. Either one. I will be as attentive as I possibly can. I'll get back to you as soon as possible. And I know I don't actually know any of y'all, and that you're just these blank silhouettes, icons behind the screen, but I've always just really felt responsible for my students. I care deeply about your progress. There's no way I could not let y'all miss a lesson. It could not happen. And I also just feel really bad that this happened the way that it did. I'm sorry. I literally found out at the beginning of the week that that the lesson was not going to be able to take place, and I've been working behind the scenes to try to get something happen, and nothing's happened. So, for that reason, really, really hoping that this is adequate, and to make up for that, any questions you have, I will be 100% available. Not that I'm not, but I'm just further encouraging. Not that I'm ever not available. I'm just further encouraging as many questions as you possibly could have to send me. Please send me. Okay. So, if you're watching now, you got this recording. You can pause or fast forward or 2 times speed or do whatever you want to do. So maybe that'll be a little bit of a benefit to y'all. But you won't. The downside again is just us interacting and being able to address your questions directly. I'm not going to be able to explain anything that I do further than I choose to myself. I'm not going to be able to say, you're not going to be able to stop and say, hey, like, can you? I'm not. So, for that reason, I'm going to be slow, very, very slow, very deliberate in everything that I say. and there will be pauses. Maybe if I need you to look up something or to be involved. Okay. It might sound like I'm Dora the Explorer when I speak and then pause for a second, but we'll just work through this. I've done some recorded lectures before, so it's not the first time. We also there will be some things that I send you on email. You would have gotten these by now, that are companions to this lesson. Okay. They're not the highest quality materials. In fact, they're just some pictures of a screen. But that's what I've got for today. The plan would have been for me to do this lesson out loud, and I could have copied and pasted this and sent y'all, I could have either had the materials shared on my screen while I was teaching, and it could have worked better. I had a better lesson plan. But because of this, I'll need y'all to open the files or images that I send you and follow along with this lesson as best you can. Okay? So, the first thing I want to say is I've realized the Anki thing, there is a little bit of trouble with that. Some things that I did not realize until just now. So, I'm going to re-explain that for people that don't know. And if some of you that do know, you can just fast forward until I stop talking about Anki, and I'll try to say that word as frequently as possible. So if you fast forward, anyway, you know what I mean. Fast forward if you're an Anki pro and you've been doing this, you don't have to do for the next couple minutes, okay? Several minutes. But Anki is the application. That is the tool, okay? It is free on the internet. It is flashcards. I hate to be a just trust me bro about it, but just trust me, the way that it knows to show you flashcards is phenomenal. It knows what you need to do. It knows if you missed a card on Friday that how many times you need to see it on Saturday, or if any. It knows that if you got this card right 4 times in a row that you don't need to see it again for several weeks. And it knows that if you miss it again after several weeks, it will then correct all the cards that it showed you that it showed you from back then and move them up a little bit further. It learns and it knows more about you than you do. Okay, it knows your middle name, it knows the name of your first pet, it can answer all the security questions on like the secure website that you need a password for. It knows everything about you. So it is very important for you to do Anki, the cards, every day. You don't have to unlock new cards every day. So if you get this Anki deck that I'm about to tell you about, you can suspend and unsuspend cards. I'm going to show you how to do that. You don't have to open new cards every day, but you have to finish whatever old cards it chooses to show you every day. Let me show you what that looks like, okay? I'm going to pull mine up. I'm going to have to hold it up to the screen. It's probably the best I can do for right now. I've thought about doing a screen recording, but the power to stitch several videos together would be too much, because I'd have to use a tool that does that, and the uploading would take hours and hours of how long these videos are going to be. And I didn't want to send y'all like several different videos. So we're going to do it all at once. You can fast forward, a little bit more low tech, but I think it'll work, okay? So I'm just going to pull it up. This is what Anki looks like. And mine is not updated, so yours might look slightly different, and everybody's looks slightly different because there's customizations you can put on there. This is Anki, okay? I'm going to scoot closer. This is Anki, all right? And to y'all, it might be backwards. I hope the video flips itself over. So, when you go to Anki, you have these decks right here. Each one of these is a different deck or a sub-deck. You can download these, click on this little arrow, and or this little sign, it'll show you the subdeck. So if you click on that, you see how it changes? It hides or shows the subdecks. All right, they're very, very, very organized if you download the ones on Anki Hub, which we'll talk about in a second. Okay. Now, if you go up here to browse, right here, okay, you click browse. Okay, these are individual cards that are on the Anki. These are the individual cards that are on the Anki. You click browse, it takes you to a new screen. You can highlight these cards just like you highlight anything on a computer, okay? So you highlight them, okay? You can left click or right click on them. Well, I right click too hard. You right click on them, and there's something on here. It says toggle suspend, okay? You can suspend those cards. When I get off of them, see now they are yellow. They're highlighted a different color. Yours might not be yellow, but they'll be highlighted now. You can tell that they're suspended. Okay. What I recommend doing, this is what everybody in med school does. Okay, this is the method. There's slight differences. You can adjust the specifics, the specs on your Anki. You can do all that. You're not going to go wrong just knowing or just keeping the basic Anki stuff, okay? Don't worry about it, all right? Okay. But suspend all the cards in the deck that you download. All of them. Suspend them, every single one when you start off. And then only open a few at a time. If you open 300, let's say open 300, okay? You're only going to have to do 300 cards that day. That's your day one. First day, 300 cards. For new cards, if you keep the settings the same, you will have to get it right at least twice. If you miss it several times, you might have to get it right more than twice, but usually just twice, okay? The more you miss it, the more frequently you will see it. So if you miss the same card 5 times, you will definitely see it tomorrow. If you get this one card right twice, you might not see it for a couple days, okay? Now, when you do a card, I'll talk about that in a second. Just suspend the cards, or unsuspend the cards as you go. So suspend all of them and then unsuspend a little bit at a time in chunks, okay? Now, let's say you get 300 cards. Let's say you get half of them right. Bam, bam. You won't see those cards for maybe 2 to 4 days, depending on how your Anki is set. Let's say you missed a couple times, once or twice, the others. You will see those tomorrow when you do reviews, okay? If you look here, you have these in blue right here and these in green over here. The green is the reviews. Those are the reviews. Those are cards that you opened up and have already seen. You've already got them right a few times, and Anki is deciding to show you them again, okay? Those cards are very, very important to do. If you don't want to open new cards that day, you don't have to. You should open a few cards a day, most days. If you want to take a break, don't. But you should always do the reviews. Even if it's an off day for you, do the reviews. If you have an important day, like let's say it's Wednesday and you know on Saturday you do not need to be attached to your phone or computer for very long, and you're one of those people who's aggressive with Anki, you might have 1,000 reviews a day. Just don't open any cards for a few days and those reviews will dwindle down. Because, like I said, if you miss something that you opened for the first time today, if you missed it a couple times, you will see it tomorrow, the next day, okay? So, open the cards a little bit at a time, and then once you start, at least do the reviews every single day, but you can time that up the way that I was just telling you. Because, like I said, you open up 300 cards, if you get 150 of them right, bam, bam, it'll be a couple days before you see any of those. But the 150 you miss once or twice, you will see very soon. So the next day, the very next day, you will have about 150 reviews. If you open up no new cards on day 2, on either day 3 or 4, you will also have 150 cards-ish. Those cards, if you get them right again, you won't see them for like a week. And let's say you get third time, on day 3, let's say you get half of them right again. So for some reason you lost a step that day, and even though you got them both right the first day, you got it right twice, you missed half of them the next day. You will see those cards again on day 4, and you will probably see some of the cards you reviewed on day 2 also on day 4. Does that make sense? So they build up. But Anki knows when to show you the cards. The only thing that's very, very, very important is that you do the reviews every day. If you don't want to do a lot more than the reviews, don't open any new cards. Don't unsuspend any new cards. Okay? You download the deck, you suspend every card, and you unsuspend a little bit at a time. They're very well organized. If you want to unsuspend them by category, if you want to learn a certain thing that day, you can go back to browse, okay? And over here, if it'll load, yes, on the left, I cannot get into all of it, but they are very, very, very well organized. And you can click on these little clocks and flags and colors, and you can figure out what the title of those cards are. Okay, the cards are very there's way too much that I could have to teach about this deck, about this application right now. But if you keep it simple, you can still accomplish everything you need to accomplish. Okay. The reason I say this is because every single piece of content that you need is probably in this Anki deck that I'm about to explain. It is, if it's anything like, and I did not use this deck to study. I wish I had, because I spent a lot of inefficient amount of time making my own cards. I made several of my own cards, a lot. And this, you don't have to do that. If it's anything like the deck for step 1 and step 2, which are phenomenal. Those are phenomenal, and they're made by the same people. So I imagine that this is also phenomenal. The decks for step 1 and step 2, if you learn every single one of those cards, you will not miss a step 2 question. Of course, nobody's perfect. You're not going to 100% perfectly learn all those cards, but they have every piece of content you need, literally from the book, from the step book. And because Anki is so good, it is the quickest way to learn the most information. It is very, very handy, very powerful. Every med student, the med students that don't use these are either kidding themselves or they are genetic freaks that can do it another way, because this has revolutionized med school so much. People have been using this long enough that we are learning more, and medical school is figuring out that they're kind of getting too good at this, so they have to kind of make med school harder. And med school has gotten harder over the past 10 or 15 years because we are speed running learning as much information as possible, and it's becoming too efficient, right? So it's fine because we're outpacing the curriculum by a little bit, but right now med students can do more. They can do more extracurriculars. They can be more competitive because they have the most efficient way to learn this stuff is pretty much this right here. It is a miracle. Okay. It's hard work because you have to do it every day, or at least your reviews. You don't have to open new cards every day, but it is a miracle. Okay, that's all about the app, Anki Hub. I'm not going to pull it up right now because my subscription and login is expired. I feel like I remember my Anki Hub subscription either being really cheap or not expensive, because I remember, because I'm very broke and I worry about money a lot, and I don't remember worrying about that. Whatever the case, I found out for the Anki Hub thing that I sent y'all, you have to either get the core or premium subscription, which is annually it's like $66 and like $102 or something like that for each one. And I'm pretty sure the core will cover it. Do the research, figure out what you do is you go to the Anking. It's like Anki, but with NG at the end, okay? Anking MCAT deck. It is verified. It is recommended by medical schools. People have worked very, very hard on it. It is regularly updated. If you link your Anki Hub account, that is an online website where you get access to these decks. That is intimately related to Anki, but it's not the same thing. The website is a tool to help you use the tool of Anki. You go on that website, you're going to have to get either a core for like $65 or for like $100, the premium subscription, one of those two. You can look up the differences. They're explained. Nobody is trying to trick you on those websites into spending more money than you need. They know. Okay, it's for a year that when you pay it, you can get that Anking MCAT deck, and it will have everything you need to know for the MCAT for all 3 non-car tests. Okay, you will subscribe to the deck. You subscribe to it. That will allow you to upload it or download it to your Anki, your website application that's on your computer. It's on on Apple. It's on my Finder. If you have like a Windows computer, it'll be like you click the window application. It'll be somewhere there, something you download. You can download decks to that via Anki Hub. You can sync your Anki Hub to your Anki app. Those are two different things that Anki Hub helps you use Anki. Okay. You also, for if you want to get the phone version so you can do more reviews, most med students end up getting the phone version eventually. I was able to, in medical school, I didn't have to get the phone version until I got about 3/4 of the way, maybe 2/3 of the way into my step 1 studying. So I spent probably 9 months studying for step 1, and maybe the first 6 months I didn't have to use on the phone. And then I started having too many cards, and I was like, if I don't get this on my phone, because again, I'm broke, I was trying to pinch pennies. If I don't get this on my phone, I'm going to be up all night trying to do these, because I can do them. I walk a mile to the school every day, to the parking lot, and then a mile back, and I can knock out 100 cards each way. Just that's not a bad idea, but don't feel like you have to do that. The web app Anki is completely free. It's amazing that we have access to something like this for free. I'm imagine that Anki Hub is just got too fed up with being a free resource, and maybe that's why they had prices, or maybe I don't remember the prices for med students, or maybe med students get it cheaper and pre-meds are out of luck. I don't know. But please use the Anki. It is a lifesaver, and I promise you, so for content things that you have to figure, and like I helped a student the other day learn how to balance chemical equations like you learned in high school chem. Anki can't help you learn that, but the fact that carbon's oxidation state is +or-4. Anki could help you learn all those things. Maybe I don't know what's on the Anki MCAT deck, except that a lot of students use it and it's successful. And because a lot of students use it, I've explained to y'all before that Anki, that the MCAT is a contest, and you have to do more, at least as much, if not more, than other students. If say 30% of MCAT students are using this, that is an advantage that they have that if you don't use it, you don't have. And so it would behoove you to keep that competitive advantage as high as you can, because it's a very good tool. Now, there are other decks on Anki Hub that you don't have to have. You can have the free subscription and get, that are not the OnKing MCAT deck. I just don't know how good those are. I have no way of knowing. So you can do your own research and find out if those are acceptable to you, and not do the OnKing and not pay the money. That's totally fine. But I just can't speak to those. The only reason I know OnKing is good is because I used that deck to study for step 1, and I took step 1 like 8 weeks before anybody in my class did, and I just used those cards and did questions. That's all I did. I really did 2 things for step 2 or step 1, and so yeah, that's that. Okay, so now we're going to go into the lesson that I wanted to teach, but I feel like that was very, very important. And for those of you that already use Anki and that was pointless, I'm sorry. But I've had some of my individual students were doing 15 cards a day, and I was like, no, that's not what Anki's for. You got to do more than that. And then some others were like, I tried the Anki Hub thing, it didn't work, and I found out about this core versus premium thing. And so I wanted to make sure that wasn't y'all. I'm going to try to pack a very, very important lesson, though, into the rest of this time. That is something to use for science passages. So, for car, y'all are doing the drills where you read as hard as you can, time it with a timer, or a stopwatch, I should say, a stopwatch. See how long it takes you, write that down. Do that several times a week. You're also doing the drills where you set a 3.5 minute timer, not a stopwatch, a timer. 3.5 minutes, read as fast as you possibly can, and then stop, and then don't read anymore. And for both of those, you're doing the matrix method. The matrix method you do not use on the real test, but it evolves into something that you could use on the real test, and that evolves into something that you could definitely use on the real test. And we will go over those soon. Probably about week 4, probably next week, we'll start the new method. So I want everybody to have done the matrix method at least 40 times before then, okay? Sound good? You can do it on the car passage I sent you, or any car passage that you want. Anything. But use it. When you use the matrix method, don't look back at the passage while you're doing it. Do it from memory. You don't have to time the method. You do it take as long as you need. It shouldn't take that long. But don't time yourself on that. I had a student think that was a timed test also. It's not. Okay. This method is a method you can use on the real test for science. Now, unlike the other methods, for the things I'm teaching for car, it is not as definitive. Part of using this method is knowing if you should use it and when you should use it. And on most science passages, you can use it, okay? It will help your thinking. It will be very, very clear. I'm going to do a car lesson soon, and my car lesson will be about taking the big picture, simplifying, looking past the reeds, getting through the brush, looking at the bare main point, very, very simplifying things. This does the same thing for science. This will simplify science passages so that you can understand and follow them, and you can do this on the real MCAT. This board is similar to what you will have on the real MCAT, so this is even better practice, okay? You can do this on the real one, and I'm going to go very, very slow, but you can do this very quickly. You will develop a shorthand, okay? And my shorthand might not fit what you decide, but you have to you can develop your own code that only you understand, because nobody else needs to see it. Nobody's going to grade this, but I want y'all to practice this method. Okay, I've sent y'all, unless if I haven't, somebody watch this video and remind me to send it. I've sent y'all a passage. It's not good quality. It's literally a picture, but you can read it perfectly fine. Like the reading is good. It'll be 2 pictures at least of a science passage that I'm basing this off of. Okay, so right now, if you have not done that, pull that up on a different device, on a phone or iPad or something. I'm sorry that I can't do like a dual screen situation here under such short notice. I'm actually not even at my own house right now, and I'm doing the best with what I got. Okay, I'm very sorry, but I'm sending you this. You can be able to see me and read the passage. Okay, I know that is not ideal, but that's what we're going to do. All right, so here's what's going on. I'm reading this passage. I'm diagramming it. You're going to see it's called the flowchart method. You'll see, okay, what I'm doing. And as I'm reading it out loud, y'all are going to be able to follow along and see where I'm getting at with my whiteboard, okay? Again, you can do this on the real test. It'll go quicker when you're not teaching somebody, okay? So for this science passage, this is a biology passage, and this is usually the most effective on Chem Phys and the least effective on Psych Soc and medium on Bio passage. But this is a great Bio passage. It works great on this one, and even when you get really good at it, some that you might not, that maybe earlier in training you're like, oh, I couldn't flowchart that. You realize you can flowchart it, and it really helps you simplify things. Okay. So I'm reading from the passage that I just sent y'all. Okay, there's 2 pictures. Excitation, contraction, EC. Okay, when they put something in parentheses, they are going to reference it again 99 times out of 100. Okay, the parentheses are there to confuse you. They would just type it out, and to save space. They do save space, but they're there to confuse you. And a lot of times, the parentheses that they put them in have nothing to do. They don't even appear close to what the symbol or the shortened version is, not close to what the long version is. Sometimes it is, sometimes it's not. You need to work in your brain right now how to see the initials as the thing that you're talking about, okay? It is important. Important to do. All right, so excitation coupling in the muscles is dependent on the cup line. A group of proteins involved in the modulation of calcium barrier between cells. Usually these science passages, the first sentence or 2 is very rudimentary background, but sometimes it can. A lot of times they're always important, but they're not going to start explaining scientific relationships just yet. Usually, this one gets right to the meat of it pretty quickly, but there's still more background in this first sentence than obviously in the sentences that follow. EC, excitation coupling in muscles, is dependent on the cup line, a group of proteins involved in the modulation of calcium between cells. Initially, calcium is stored in the sarcoplasmic reticulum by interacting with the cup line calsequestrin, a calcium binding protein. Okay, you can diagram this. Okay, hopefully most of these science passages, if you got a great content background, should be a little bit familiar to you. They have every right to write about something that you could have never heard of, that nobody should have, that studying for the MCAT could reasonably have heard of. They can do that, most of the time it is something you've heard of, and hopefully y'all are familiar with how muscle cells work, at least a little, okay? You've had biology class. This is not completely foreign to you, but even if it is foreign to you, you can still do this method, okay? Okay, so calcium, I'm going to write a little bit bigger than I normally would. Excitation coupling, you've got calcium, and I'm just going to draw that as Ca2+, is stored in the sarcoplasmic reticulum. Okay, so this is what I've got right here. It is stored, there's calcium inside the sarcoplasmic reticulum, okay? And if you wanted, you don't have to make it Ca2+. You can make it, you know, maybe you're shorthand, you're like, okay, I know calcium is ions. Ions are like atoms, so I'm just going to draw little dots. And I know that that's calcium. If that's how you want to do it, that's how you want to do it. I'm going to do both. I'm going to have the Ca2+ on here. Okay, can everybody see that? Ca2+. Okay. So, stored in the sarcoplasmic reticulum by interacting with the couplon calsequestrin, CSQ. Okay, CSQ is calsequestrin. So, calsequestrin does this somehow. It somehow, don't know how yet, it stores, or it causes calcium to go to the sarcoplasmic reticulum, okay? This is a good key here. Sequester means to hold away, to put away, to keep separate. That's what that means. So keep thought there. It keeps calcium away in the sarcoplasmic reticulum. You don't have to know that to get anything right about this passage, but it helps. Helps you conceptualize. If you know, good to know. Always good. Never bad to have a big vocabulary. Okay. Okay. During EC. So during EC, we already have EC written, excitation coupling, in muscles. Membrane depolarization triggers calcium release from calsequestrin, leading to muscle contraction. Okay, so seems like that sentence makes it sound like calsequestrin is what is actually holding the calcium more than like the sarcoplasmic reticulum. That's fine. So all you do, if that's happened, if your thought process changes a little bit, just connect these two things. That's all I'm doing. So now calsequestrin is a thing that is on the sarcoplasmic reticulum that's holding the calcium. Because that's what this next sentence kind of makes it sound. You don't have to think too much, okay? So, during excitation contraction, you release calsequestrin here. So, you can put EC, several things you can do. You can draw lines here, which I like to do. You could also say EC equals calsequestrin minus Ca2+, because that's what it says. Excitation coupling, calcium is released from the calsequestrin. That's what I've got right here. Sounds good? Okay. That's what leads to the retraction, to the contraction. Calcium reuptake by another couplon, the sarcoplasmic endoplasmic reticulum calcium ATPase. That's a big word. Luckily, they're going to call it a good acronym, CIRCA. When you're reading, I've been trying to read EC as excitation coupling so that I'd keep it straight, and calsequestrin is calsequestrin, not CSQ. That's a big word. So try to just use the acronym for that one in your head, but make sure you know what the acronym stands for. Don't read each letter of that acronym as you're going. You're going to waste a lot of time. Okay? But you have CIRCA. So, relaxation. So this is excitation coupling. We're going to do another math problem. It just said relaxation. I'm going to reorganize this in a minute. And I'm going to say MR for muscle relaxation. Okay? Just for time. Equals CIRCA plus calcium. Okay? So here we go. This is where that's written right here. Okay? All right. On your own thing, no, this isn't like super lined up well, but when you're writing yourself, you'll know where everything's at, right? So I already wrote this excitation coupling equals calsequestrin minus calcium. Muscle relaxation equals circa plus calcium, because it says calcium reuptake by another couplon leads to the circa leads to muscle relaxation. Circa is regulated by sarcolipin, SLN. So that's an easy one. When I like when something's modulated by, I like to attach it. So it's attached right here. Okay, and because I also made an attachment right here, you might want to just draw like an arrow here to let you know that it put a little M there, modulates. Okay, if everybody can see what I wrote there, this little M. All right. Okay. So do y'all see where I'm going with this? You're trying your dangdest, your darndest to make this mental picture in your mind to simplify the passage. And I'm going to draw, I'm going to just reorganize my thoughts so it's better for y'all. I don't have the best handwriting as I'm still doing this. You're feeling this out though. You're not, if you get to a point and you're like, okay, that doesn't make sense, or the drawing that I've got here doesn't really help me explain that, you can stop doing the flowchart and just read if you need to. But these passages have a lot of big words in them. They have a lot of stuff to confuse you intentionally. They actually take science, they take real studies for these passages. They strip them down of all their like specifics, and then they add in more stuff that is technically understandable but is intentionally confusing. Stuff that you don't have to have a background to understand, but they could have made it, if they were just trying to inform you of something, they could have made this a lot simpler. They're not trying to inform you, they're trying to trick you, right? So don't be too, this helps you be less confused by these passages. These passages are designed to confuse even smart people, okay? Very, very easy to fall victim to this, and this just lets you keep it simple, okay? You might get to one passage on there one day that you just get naturally because maybe it's in what you did research in, or maybe it's what you're interested in. And maybe you're like, I don't even want to take the time to do the flowchart because I just get it so well. But if you start doing this now, you'll still have time. You'll be surprised how quick this takes you. You can organize your thoughts and refer back to this instead of referring back to the passage so much and having to hunt through. This is like a cheat sheet, okay? Very important. All right. I'm also drawing here the sarcoplasmic reticulum, which is intimately bound to calsequestrin. So drawing an arrow from calsequestrin to sarcoplasmic reticulum, which holds calcium. Okay. So if y'all see what I've got here, the same math problems, okay? Excitation coupling equals calsequestrin minus calcium. When basically restating exactly what the thing said, when calsequestrin releases calcium, you get excitation coupling. Muscle relaxation occurs when circa reuptakes calcium. Circa plus calcium reuptakes, or circa reuptakes the calcium. Circa plus calcium, muscle relaxation. SLN is the sarcolipin that I just put that. All it says is that it modifies it. It's regulated. Doesn't say anything else. If you want to put a little arrow here and put like an R for regulate, that works. Make it capital. But you don't have to do that. Like that just is extra. And then of course at the very beginning we talked about how calsequestrin is in the sarcoplasmic reticulum. Here's the sarcoplasmic reticulum with dots and calcium ions in it. Okay, everybody can see that. On the actual thing, real life, this would probably take you an extra 17 to 25 seconds to draw. Okay, but now you took those extra seconds. Now you don't have to go back and read the first paragraph when you do questions. Now you have it all. This is the entire first paragraph of this passage right here. Okay, very important. All right, so now we go to the second paragraph. As I zoom in a little bit here, turn my brightness up so I can see. Okay. Triidin, another couplon encoded by the TRDN gene, is a 95 kilodalton transmembrane protein in the sarcoplasmic reticulum that modulates blah, 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 blah. Okay. Pause. There are some details in there that we don't know if it needs to come up yet. the so far nothing about genes or the kilodaltons, which is the size. That's how you measure proteins, is in the mass of them. Okay, none of that has been mentioned yet. If you're just learning, you might want to try to jot something down about that. However, I've done this a few times. This is a tip. If they completely shift gears in the science passages, just hold up a second. Okay? Maybe don't write that down until you realize it's important. Because if I have to read the rest of this paragraph, you're going to realize that Daltons nor genes comes up. And we're not going to do the questions tonight, but I'll give you a hint. There is no, we'll do a couple of questions maybe, but there's no genes or Daltons in the questions. They're trying to trick you. That's why you do the flow chart. Just like on the car, when you only write down the main idea, the main points. I don't want you to write down details. I want you to write down, right now, arguments, which should basically be summary in your matrix method. You're writing down author, tone, all thinking about big picture, why the author wrote this, what energy does this whole entire passage give off to you. The argument section should probably be main ideas of individual paragraphs, and the main idea, of course, is the main idea. I want you to think big picture, okay? But this helps you think big picture of science, helps you simplify science. That is a detail that if you get caught up in, you're going to get lost. Right now, the point of that in being in this passage right now, the gene and the kilodalton thing, and it's going to work on everybody that reads it, because your brain's going a million miles an hour. You have to consider everything and be vigilant. But right now, the purpose of that is to make your brain think about something irrelevant. It wants you to start thinking about the size of proteins, genetic code, residues, like folding, genes, ribosomes, translation. That's the kind of thing you might be popping in your head when you see transmembrane protein, 95 kilodalton, TRDN gene, encoded. I see the word encoded and my brain just completely wigs out, because I'm like, oh my gosh, I'm going to see something about like first semester biochem when there's ribosomes and DNA transcriptase and RNA and all that. None of that comes into play here, okay? So when they switch gears real bad, or if they've mentioned something that's not really in line with what you've done so far, hold up, keep going, and make sure it's not important, if it's important, because here it's not. So I'm going to keep going. Triadin, TR, is another couplon. And if you wanted, they've said some of these are couplons. If you want to go in here, because like the sarcoplasmic reticulum is not a couplon, but the circa is. If you want to put like a little happy C, like to label the things that are couplons, you might want to. That might help you keep up with what's a couplon and what's not. That's just an option. You don't have to do that. But you see here, I put these little happy C's by the things that happen to be couplons, okay? I'm going to try to keep this held up so y'all can see it. So triadin, another couplon, encoded by the chain, blah, 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 is a transmembrane protein of the sarcoplasmic reticulum that modulates calcium signaling for EC coupling of skeletal muscles. Okay, so there's electrical contraction again. So it is in the sarcoplasmic reticulum, okay? It says in the transmembrane, yes, in the sarcoplasmic reticulum. So I'm going to draw it here. I'm going to put a happy C on it because it's a couplon. It's called TR, triadin. Triadin, we know, modulates calcium signaling for EC, for the excitation contraction coupling of skeletal muscles. I would read further because the next sentence that starts with the word TR, but you already might want to just, if you're drawing this and you're working fast, draw like an arrow to this relationship that we made right here, excitation coupling. This is excitation coupling right here. We drew an arrow to it. I'm not going to do that just yet, but you might want to. Okay? Triadin anchors calsequestrin to calcium release ryanodine receptor, Ry1. Okay? Ryanodine receptor. So TR anchors calsequestrin to ryanodine receptor channels at the junction with transverse tubules, the enfoldings of plasma membranes. Figure 1. Okay, there's a picture here. I don't want y'all to redraw pictures. If the picture is there, do not try to redraw it on here. The picture's there for a reason. The pictures help simplify things themselves. We are going to illustrate these relationships real quick, but we aren't going to redraw. Don't reinvent the wheel, especially if they're like a chart or a graph, like a bar graph or a line graph. Do not draw that on here. Don't draw diagrams on here. They're there for you on purpose, okay? They're not going to lie to you, and you don't want to waste your time trying to draw something that's already here. I'm going to illustrate that relationship a little bit just for thoroughness sake, okay? But don't just try to reinvent the wheel by drawing the same thing that's there, okay? So, Triadin anchors Castle question. So now, the thing about this is you're going to end up drawing lines through your drawings because you don't know where how to space them out. You could make it look nice and pretty. So, I'm going to erase a little bit here so that I can make it look nice and pretty for y'all. The good thing about this not looking nice and pretty is because when you're doing it yourself, you know how your brain works. You can kind of see it. It doesn't have to look good. When I'm teaching, I want it to look good so that y'all can tell what's going on. Okay. Okay. So, Triadin anchors calsequestrin to the sarcoplasmic reticulum. So I just like to draw two lines when they say like link or join. Okay, so Triadin anchors calsequestrin sarcoplasmic reticulum. That's what we're doing here. I'm sorry. Triadin anchors it to the ryanodine receptor. This is what I get. Sorry. Anchors it to the ryanodine receptor, which is Ry1. Gotcha. R1. Sorry. Triadin anchors calsequestrin to the ryanodine receptor. I'm sorry for the confusion there. So here we go. Triadin anchors calsequestrin to the ryanodine receptor. And you understand the way that relationship goes in your head, right? In your own head. You would draw this slightly different. Everybody has their own things that they want to use, okay? Then they explain how the anchoring works, and that might come up, it might not, but I'm not going to draw that because it's drawn for you. Okay, so that's why I'm not going to get into all that. Okay. L-type voltage-gated calcium channels in the T-tubular membrane undergo conformational changes upon depolarization and trigger nearby Ry1s. See, they kind of changed their acronym here, and that's why this is not a real MCAT passage, so they would not change the acronym this on a real MCAT passage, but they changed it a little bit. Trigger nearby ryanodine receptors to open. The alpha-1 subunit of Chi1 is responsible for voltage sensing. Okay, so a ryanodine receptor right here, okay, basically another protein that we're talking about here, and it's called, they abbreviate it CAV, but they're L-gated, voltage-gated type calcium channels. This is what I talked about. Some of these abbreviations make no sense. Okay, and that's not a. That might not be a V, or it might be a Greek letter. I don't know. With an alpha subunit, alpha-1 subunit. This thing right here, the voltage-gated channel, triggers Ry1 to open. And you can write open right here. Something like that might make it easier. Okay, do y'all see what I've done there? CAV, the voltage-gated channel, calcium channel, triggers Ry1 to open. Okay? All right. In addition, now in the next paragraph, it gives figure 1. In addition to anchoring Cal1, Triadin also plays a role in the expression of other couple-line proteins. To study this, so when it says plays a role or something like that, look out for non-specific words. If it's not something like regulates, modulates, upregulates, increases, decreases, if it doesn't describe a relationship, A, they might be about to investigate that relationship. That's what's going to happen here. B, it's vague, don't write it down. Don't feel like, oh, Triadin plays a role, unless it says it plays a role that results in this and it explains it, but be on the lookout for vague language, okay? So, in addition to anchoring calsequestrin, Triatin plays a role in the expression of other couple-line proteins. To study this, researchers rendered TR non-functional in cells through ablation. Figure 2 shows the effect of triatin ablation in the expression of couple-line proteins. Okay, that's the end of the passage. So you're done drawing because now all you have left is a bar graph. You're not going to redraw the bar graph, okay? So I'm going to hold this up. If you need to pause the video and compare this to what you might have been drawing if you were drawing, or just to pause this, take a screenshot, do what you need to do to see how I got to here by reading the passage, please do so. I'm going to give you several seconds to do that if you need to do that, okay? I'm going to go through and label everything. Excitation coupling happens when calsequestrin releases calcium. Muscle relaxation happens when SERCA reuptakes calcium. It is modulated by this protein right here, sarcolipin, regulated by, okay? The calcium is stored in the sarcoplasmic reticulum by calsequestrin. That's where it stores it. Triatin is also in the sarcoplasmic reticulum. It anchors these channels, the rhyodidine receptor channels. Triatin anchors rhyodidine receptor channels to calsequestrin. When the voltage-gated channel opens up rhyodidine to let calcium flow out. And I probably should have drawn the calcium flowing out. That's what's going on here, okay? So there's a short explanation. I explained the whole passage, just from that right there. Now you know everything you need to know about this passage except for what's on the bar graph, okay? Okay, so we go to this bar graph, and I hope all of y'all are looking with me on this bar graph. Actually, I'm just going to hold it up to y'all. Let me see if we can get it up here, okay? I'm going to zoom in. We're going to do that. I think that would work best for everybody. Okay, here we go. So, on this graph, you have read the X and Y axes of all the graphs. Read the captions of all the graphs and the figures. Very important, you have to know what is being measured, okay? What is on the bottom of these? If you read the paragraph, this very short paragraph before this, what's on the bottom of these is the different couple-line proteins. They're on the bottom, okay? What's on the side? Relative expression. It does not explain that anywhere. This is a case where you need to know what the word relative means. That's not a science word. That's not a, it is a science word, but it's not anything above high school freshman level word. It's something that y'all should know. Basically, you go from the reason the bar is at 1 is because a relative expression of 1 would mean unchanged to normal. Relative means compared to something else, okay? If something is relative, it means it depends. It is relative to... So, for example, if I'm 5'7", I'm not tall, but I am tall relative to people with dwarfism, okay? Again, this expression that they're giving you is relative to their normal expression in a wild cell, okay? Because these are the triatin knockout cells. You see how triatin is on here and there's nothing, triatin is right here and there's nothing there. The triatin's gone because these are different proteins that are listed. Now, personally, this is a slightly misleading graph because the way that they describe the parameters, triatin should not be a thing on the graph. It shouldn't be. Basically, your dependent variable is on the line where your independent variable is on the line where the dependent variables go. So I don't like that. Dependent variables go on the x-axis. Y-axis is usually your independent variable. Dependent variables are what is measured. Your independent variable is what you did to the program, to the experiment. I can explain that better in another lesson. I won't have time tonight. So if you struggle with that, please ask. That could be an important MCAT topic. Not specifically an MCAT topic, but it might help you understand some science passages. But basically, in this graph, the outcomes are on the X, and what happened is on the right, is on the Y, okay? So, when triatin knocked out, what happened to these other proteins? It said that triatin affects the expression of other proteins. This first one, calsequestrin, it went down, because relative would be 1, Okay, it went 1 out of 1% or 100%, as in it expressed the normal way, versus it's closer to 0. Not no expression, but less expression. Okay. CALSA question expression went down. Do you see the star here? That means significance. You notice there's not a star here. You cannot say anything happened to this protein right here, which I believe is sarcolipin. Okay. There is no relationship between triatin and sarcolipin because there's no asterisk here. It explains in the caption that the asterisk, you have to pay attention to this, means statistically significant. And if you don't know what statistically significant means, email me and I can explain it, okay? So, there is a relationship between triatin and calciquestrin. If triatin's gone, calciquestrin goes down, okay? Sarcolipin, no relationship. Circa, there is a star here. It is above this line. It goes up. This is the the L-type voltage-gated calcium channels. Those are also expressed more when triatin is knocked out. And so are the ryanodine receptors. They are expressed more when triatin is knocked out. Okay? So that is how you interpret this graph. This is not the best graph. This is from Kaplan. I have been investigating these Kaplan tests. I think they are very useful for people to take for a couple reasons. Stamina, if you're trying to practice stamina and you want to take a full test, you can sit down and take that test, and you will know that you can sit through an 8-hour test. Number 2, you can learn some content from the tests. The Kaplan tests are not very diagnostic. They are very much very content-based, way more content-based than the actual test. And I'll swear by that. I've seen several Kaplan passages in a row now. I've researched this myself and seen what other people think. I've reached out to my other tutor colleagues, and they have also kind of agreed with me that, yeah, some of these Kaplan passages are very content heavy. They are not realistic MCAT choices. So if you're using a Kaplan test, first of all, your flowchart method is not going to help you much on content related questions, which we've explained is that when I say a content question, I mean a college-level science class is needed for you to get that right. I do not mean that you had to know something on the periodic table to get that right, because A, you have a table on the test. B, you should have learned that in junior year or senior year of high school. I don't mean that you can't know any science at all. You might have to use junior high or high school science. I had to help a student the other day balance an equation, the equation for photosynthesis, carbon dioxide plus water equals glucose plus oxygen. That is high school chemistry, okay? That you might have to do something like that, okay? But 90% of the questions will not have a college level science slant to it, okay? That is why I do the flowchart method, because the passages are designed to confuse and confound you. The flowchart method helps you simplify things and find the relationships, because these passages are all about relationships. Now, the Kaplan test, there are way more than 10% of questions that you kind of need science-related. You need college-level science. For example, the first question on that passage was about which of these very unique cell features do cardiac myocytes have to help communicate with each other? Has nothing to do with the passage. The answer is intercalated discs. We learned that in med school. Like, that's high school science. You get a good high school science class. Okay, that's not. If there's one of those per passage, okay, but it's a lot of passages are like that. But the next question is very, very simple. Let me see. And here's the thing. I'm not going to read the question to you because you don't have it in front of you. And I'm not going to send you like 15 pictures and screenshots to confuse y'all. But this next question, it says, students hypothesize that the muscle relaxation might be affected by the size of the motor unit pool. Is this reasonable? The way these answer choices go, they go no because this, no because this, yes because this, yes because this. It's analyzing these relationships. You can, in all the answer choices, they name specific of the proteins that are in the passage. The right answer is the only one where the relationship was right. Where it says the no, yes doesn't matter. It was no because this, and they describe an incorrect relationship. No because this, and they describe an incorrect relationship. Yes because this, and they describe what they talked about in the passage. One of the arrows that we drew, it was actually about how is about how this thing right here, the voltage gated channels, help the ryonoidine receptors open. That was the correct answer. And then it's yes, and then another incorrect relationship. The no's and yeses meant nothing. This hypothesis is not. There's not enough information for you to say yes or no to. We don't know. We don't know enough to answer yes or no to this question. But the right answer had the relationship that was described here, and that you simplified it. You had all these proteins here that if you didn't write this down, you would have had to read this. Okay, let me go see. Is that what? No, I think the sarcoleptin. It just modulated or regulated. Maybe not that one. It takes a lot longer. When you have it written on down right here, you can just look at it. Oh, the voltage gated channel, it opens the ryonoidine receptors. Boom. So that can be a plausible answer. And you can see all these other answers are not plausible. You can take this flowchart and interrogate each answer choice. Okay. This next question sounds like it's going to be based on the flowchart, and then it's like asking you to name the order in which it literally says which pathway illustrates the sequence of cellular compartments traversed by newly synthesized TR as the way they move to the SR. Nothing to do with the passage. It's just asking you about protein transport, right? Where proteins get made in the cell and how they get to where they need to go. And number 2, that is a high-level science question. That is a cell bio question or a biochem question, depending on what class you took it in. And that, again, having 2 of those on one passage is very, very odd. But the next question is very similar to what I just described. They have loss of TR, loss of TR, loss of TR, loss of TR, and then relationships. You have to read the bar graph. So we interpreted the bar graph together. I hope if you need help interpreting graphs, we can go over some together another time. That is where that question came from. And then there might have been one more question on here. Let's see, maybe not though. That might be it. No, that's all the questions. Okay, so that was all the questions for those passages. 2 of them, of the 4 questions on the passage, 2 of them were completely... You can have some of those on the MCAT, but not many. And to have 2 in the same passage is not going to happen. So I just wanted to throw the little aside there. And I've seen this on multiple passages. It's not just this one from Kaplan. That the Kaplan tests are way more content heavy than the real tests. And that's okay. That happens. They're still good to take if you need some practice. Okay. So I'm sending this to y'all. It should be a link that you click to watch this. Okay. I'm sorry that this is the best I have for right now. I tried to find a substitute. They didn't work with me. They sent me, I seriously think it was a robot response disguised as a human, because it was like it wasn't even reading what I was saying. I tried to do the link that I was supposed to fill out. It didn't happen. It wasn't there. I sent screenshots to the website people to tell them, hey, as y'all can see, the link is not appearing. I need help with this. And nothing has happened as of 11:30 on a Friday night, okay? So I spent some time on my day off doing the best that I possibly could under these circumstances. I hope that it was enough. I really hope that y'all get something out of this. I really, really hope that y'all feel free to text me or to email me at any point, group me, message me at any point, and I will get to you as soon as I can with questions. I'm not going to be able to get compensated for this, but I just did not want y'all to miss a lesson entirely. So we had this lesson, again, a little bit shorter than we're used to because I don't take time to call and respond, but because of that, I'm going to be as diligent as I possibly can on some emails responses. I'm sending this now. You can fast forward it, pause it, keep it, delete it, do whatever you want with this video, so that'll be an advantage to you. And again, feel free to email me anytime. And if this is really bad, I'm sorry. I will try to do better next time. Please still leave a positive review if they have that, if that is an option on the Varsity Tutor thing, because I'm having to deal with them. I don't think they're like upset at me or anything, but the situation that happened was completely out of my control. And I'm doing the absolute best that I can. And maybe they will be more lenient with me if y'all like me. If y'all don't like me, then don't just send me an email and cuss me out or something. Don't leave a bad review. Please, I'm doing my best, I promise. Okay. I'm proud of y'all. Y'all are going to be so good. You're going to knock this test out of the park, and I'm very, very excited about the progress you've made already. And I hope that you will continue being very diligent with your studies, because I know you all have it in you if you can just find it, and that you're all going to be phenomenal, phenomenal physicians. I cannot wait to work with y'all someday, to be colleagues and peers with what as I've ascertained so far, as a bright group of students. Okay, I really, really am excited, and I promise to continue to put my heart into this as much as possible, because I want to have good colleagues like y'all, because I believe that y'all are deserving of the honor and the privilege and the responsibility of becoming a physician, and I think that y'all would be very, very well suited to that calling. So I will see y'all next Sunday. Should still be good. I went ahead and preemptively took that day off so that I'm not getting in this situation again. So I will teach y'all again next Sunday. That should be, I believe, April 27th. Okay. Happy Easter to those of you that observe, and best of luck on your studies from here forward. Thank you.